Okay, so sorry for the interruptions to the previous video. Okay, right. So we were in the process of discussing the formation of an actin filament from two actin protofilaments. Okay, so remember, an actin protofilament is this polymer of actin monomers, okay, that is this right-handed alpha helix. And now what you're going to do is entwine two of these right-handed alpha helices together. So both of them are going to be right-handed alpha helices. Okay, so here's one here, and then we can only see the back of this one here. So I'll colour one in blue and one in a different colour. Okay, so here is one of these actin protofilaments here. And you can see that it is going to be a right-handed alpha helix. So we're currently at the back here, and then we're coming forward, and then we're going to go round again, basically. So I'll put another one of these here, and this will be at the back, okay, like so. Right, then let's colour this other one in here. So at the moment, this one's at the front, you see, when the blue one is at the back, and then it's now looping forward now, it's looping backwards, okay, and then it will go behind this one here. Okay, so it's now behind the blue one, and then it will come back out of the page here, and then it will go in front of this blue one that I've mistakenly drawn here. Okay, so both of them are right-handed alpha helices, and they're intertwining together, okay, like so. And this structure now is known as an actin filament. Okay, and these are one of the key components of the cytoskeleton of cells. Okay, these intertwined actin protofilaments, which are both these right-handed alpha helices, where you've entwined two of them together. Okay, right. Now then, how are our classical cadherins going to anchor two actin filaments? Okay, so let's study this then. So, let's draw a picture here. So we'll have our cell membrane once again here. Okay, we've then got our membrane spanning alpha helix of our cadherin protein here. Okay, then we've got our 5 extracellular cadherin domains here. Here are 2 of them. Here's our 3rd one here. Here's our 4th one, and then our 5th one over here. Okay, and then we've got the amino terminus over here. Right, so let's colour all of these in. So this is an extracellular cadherin domain, this is an extracellular cadherin domain, another one, and a final one. And remember, all classical cadherins have these 5 extracellular cadherin domains, and in fact, all cadherins have at least one extracellular cadherin domain. Okay, and they can have up to 34 of the things. Okay, right. And remember, the N-terminal extracellular cadherin domain will be involved in the bonds to the other N-terminal cadherin domain on the other classical cadherin protein here. Okay, so this is also an extracellular cadherin domain. Right, now we're going to turn our attention to the cytoplasmic tail, okay, which was previously ignored, but now we're going to turn our attention there. So here is the carboxylic acid terminus. Now, certain proteins are going to bind to the cytoplasmic tail of our classical cadherin protein here. Okay, so what are these proteins that are going to bind to the cytoplasmic tail of our classical cadherin? Okay, well, there are 2. Okay, we'll start by drawing this one here, and we'll colour this one in red. So this protein that I'm colouring in red here, this is a protein known as P110 catenin. Okay, so this is called P110 catenin, and this is going to bind to the cytoplasmic tail of our classical cadherin protein. Okay, right. Now, the other one that's going to bind to the cytoplasmic tail is a protein known as beta-catenin. Okay, so this is called beta-catenin here. And I'll colour in beta-catenin in turquoise here. Now, beta-catenin is going to bind to another protein that is then going to bind to actin. Okay, right. So, this other protein is now a protein called alpha-catenin. Okay, so this one here is called alpha-catenin, and this is binding to the beta-catenin, which is bound to the cytoplasmic tail of our classical cadherin protein. So I'll colour in alpha-catenin here in vivid purple. Okay, right. So the alpha-catenin then is going to bind directly to actin monomers that are within an actin filament. Okay, so I'll draw my actin filament here. So here's one of those protofilaments, which remember is this right-handed alpha helix. Okay, and then we'll have another protofilament intertwining with this like so, to make, that should go at the back, to make our full actin filament here. Okay, so we'll colour in the actin monomers in orange here. Okay, so this is how these cadherin proteins attach onto the actin cytoskeleton, basically, and hence how these junctions that we are forming between two cells are actually going to be made stable, basically. Okay, right. So, now what I want to discuss is the fact that these junctions are going to cluster together, and that cluster of cell-cell interactions that are formed by these classical cadherins is going to be known as an adherens junction. Okay, so I just want to discuss what an adherens junction actually is. Okay, right. So let's draw two cells here. Okay, so we'll have one cell here, okay, and then we'll have another cell here. Okay, so basically you do not just have these junctions between two cadherin proteins in isolation. Okay, so you're not just going to have one cadherin protein here, one cadherin protein on the other cell, and that's it. Okay, and I'll just colour these cadherin proteins in vivid purple here. Okay, in reality you have loads of them clustered together, so you have another one here, another one here, another one here, like so. So you'll have absolutely loads of these junctions all in a cluster together, and it's often compared to a piece, well, two pieces of Velcro basically attaching in to form quite a strong adhesion between those two cells. Okay, and remember the cytoplasmic tails of these cadherin proteins, they're going to be attached to the actin filaments of the actin cytoskeleton here. Okay, and that's how each one of these is a viable junction between the two cells. Okay, right, and the name for this entire collection of cadherin interactions that you have here, that is then called an adherens junction. Okay, so we would say that these two cells have an adherens junction between them, and this means that they have multiple of these cadherin interactions that form between these two cells. Okay, right. So that then now is our discussion of the classical cadherins finished. Okay, we're now going to turn our attention to the other family of cadherins that I said we were going to have an in-detail discussion of, which is the desmosomal cadherins. Okay, right. So let's now discuss the desmosomal cadherins. Okay, so the desmosomal cadherins, they also are going to be involved in forming cell-cell interactions. Okay, so we're also going to have direct interactions between cells. So we're going to have one molecule on one cell, okay, another molecule on our second cell, and these two molecules are going to interact to hold the two cells directly together. Okay, so it's not going to be a cell matrix cell interaction, it is going to be a cell-cell interaction. Okay, right. And again, these interactions between two desmosomal cadherins, they're not going to be in isolation, i.e. you're not just going to have one cadherin on one cell, one cadherin on the other cell, and that's your junction. No, you're going to have them clustered together, okay? And the clusters of desmosomal cadherins are then called desmosomes. Okay, right. Now, we will come to the structure of desmosomal cadherins in a moment, okay? However, they have very similar structures to the classical cadherins that we've seen, i.e. they too are going to have a single membrane spanning alpha helix, a cytoplasmic domain, and then also 5 extracellular cadherin domains, okay? But the main difference between the classical cadherin proteins and then the desmosomal cadherin proteins is that the classical cadherins are going to bind to actin filaments within the cytoskeleton, as we've just seen, whereas the desmosomal cadherins are not going to bind to actin filaments. They're instead going to bind to intermediate filaments within the cytoplasm of the cell. Okay, and specifically, a good example of an intermediate filament is keratin. Okay, so I want to discuss what an intermediate filament actually is. Okay, because there are other intermediate filaments other than just keratin filaments. Okay, so we're now going to have a little study of what an intermediate filament is. And the example always to have in the back of your mind is the example of keratin filaments. Keratin filaments are all intermediate filaments. Okay then, so what's the characteristic then of intermediate filaments? Well, again, they are not just one protein. They are huge, great structures that are made by joining many, many proteins together. So let's start off with the structure that the monomers of these intermediate filaments have. Okay, right. So the monomers are all going to have very large alpha helical structures. Okay, so let's start off with that. So here's the amino terminus of the monomeric polypeptide that's going to make up an intermediate filament, and then we'll have the alpha helix here. Okay, so then we've got our alpha helix of our protein like so, and then it will end with the carboxylic acid terminus here. Okay, right, so from now on, I'm not going to draw the alpha helix actually out. I'm just going to denote it as this blue line. So the blue line will now represent the alpha helix. Okay, so the monomeric proteins which make up an intermediate filament will have a large alpha helix within them. Okay, so the proteins which make up keratin will look like this. Okay, but there are other types of intermediate filaments, and those will have, different proteins which make them up, other than the keratin filaments. Okay, which means they'll have different sequences of amino acids, but they will still have this conserved great alpha helix. Okay, right. Now, what's going to happen is these two monomers are firstly going to associate into dimers. Well, actually, we're going to take two monomers and we're going to associate them into a structure, a dimeric structure. Okay, and this dimer is going to be a coiled coil dimer. Okay, so it's often referred to as a coiled coil dimer. Now, what does that mean? Well, basically, what we're going to do is take two alpha helices and intertwine them. Now, we're not going to do this like we intertwined the alpha helices of the actin protofilaments. Instead, the way we're going to do this is we're going to form a coil where the alpha helices are kept separate, basically. So this blue line, this now represents the alpha helix. Okay, but effectively what you're going to do is you're going to wrap the whole alpha helix round in another alpha helix, like so. Okay, so this blue line, this represents the alpha helix. Okay, so in this blue line, you have got this alpha helix going round and round and round and round, but you are now wrapping it into a bigger alpha helix, basically. You're now viewing that alpha helix as being one piece of string, if you like, and you're now wrapping it into a bigger alpha helix. Okay, and you are going to coil this with another alpha helix, okay, which is made up of another protein that's already an alpha helix. So you've taken another one of these proteins. So this blue line again represents an alpha helix, like so, and you're coiling the two alpha helices around each other by making them into alpha helices and then intertwining the two alpha helices, just as we did in the case of making an actin filament where we were intertwining those two alpha helices. Okay, and this is known as a coiled coil because we have taken something that is already a coil, the monomer here, and we have coiled two of them round one another. Okay, so therefore it's called a coiled coil domain. Okay, so we'll have the amino terminus over here, the amino terminus over here as well, and then the carboxylic acid terminus over here. Okay, right. So from now on, I will just denote this like so. Okay, So I'll just show these blue lines to represent those alpha helices. Okay, and then I'll just put from now on an N here to represent the amino terminus, and then a C here to represent the carboxylic acid terminus. Okay, right. Now what can happen is these dimers that we have formed can assemble into tetramers. So you can take two of these dimers and assemble them into tetramers now. Okay, right. So what you'll do is you'll take one of these. So let me firstly draw that out again. Okay, so here's one of these coiled coil dimers now. Okay, and then what you're going to do is you're going to take another one, but you're going to put them anti-parallel. Okay, now what do I mean by that? Well, the N terminus of this coiled coil dimer is at this side, and the C terminus is at this side. The second one that you are going to take, firstly you're going to offset it by a little bit. Okay, like so. So they're not going to sit perfectly alongside one another. They're going to be offsets very slightly. And moreover, the C terminus of this one will be at this end, and the N terminus of this one will be at this end. Okay, right. So this is our tetramer now. Okay, so what are we now going to do? We're going to take 8 of these tetramers. Okay, so we're going to take 8 lots of these and assemble them into a bigger structure. Okay, right, so I'm going to reduce down this picture further. I'm now going to show a dimer just as a turquoise line. So this turquoise line represents this entire thing basically here. Okay, and now we've got the tetramer shown just as these 2 dimers offset like so. Okay, right, now what are we going to do? Well, we're going to associate 8 of these tetramers together. Okay, so here's one tetramer. Okay, and I know they look like dimers, I'm sorry about that. They do now look like dimers, but you've got to remember that each one of these turquoise lines contains 2 of the individual monomers, so each turquoise line is a dimer. Okay, right, so now I've got 2 tetramers together. There's 3 tetramers, 4 tetramers, so we're halfway through, 5 tetramers, 6 tetramers, okay, so let me move this up, 7 tetramers, and then 8 tetramers here. Okay, right, so we've now assembled one of these structures, and what we're now going to do is wrap this round in a tube. We're going to connect this one to here, okay, and what we'll end up with is a tube like so, a short little piece of tube, okay, like so, okay, where we've got these tetramers making up this tube. Okay, so here's a tetramer, here's another tetramer. Okay, and then we'll have another one going round basically. Okay, so it's assembled into a tube with a gap down the middle. Okay, and then what you're going to do is you're going to assemble this with another one of these little monomers of this tube to make a bigger and bigger tube basically. And this now, this is what's known as your intermediate filament. Okay, right, so all intermediate filaments have this same basic construction where you build a monomer which has an alpha helical structure within it, and you assemble them into these coiled coil dimers. You then assemble the coiled coil dimers into tetramers. You take 8 of these tetramers to make a tube, and then you assemble loads of the tubes together to make a huge great filament basically. And keratin is built like so. Okay, there are loads of different types of keratin because there are loads of different monomers for keratin. Okay, so you can build different types of keratin by using different monomers to make up the whole thing. Okay, but there are other intermediate filaments beyond just keratin. Okay, and these are also very important components of the cytoskeleton. They're not quite as important as actin, but they are up there. They're very, very important. Okay, now some cells have huge amounts of intermediate filaments within them. The obvious example is the cells of the skin, which have a huge amount of keratin within them. Okay, right, so that's what an intermediate filament is. So these are what the desmosomal cadherins are going to attach to. They're not going to attach to actin filaments, they're going to attach to intermediate filaments. Okay, so now let's get another piece of paper and discuss the structure of these desmosomal cadherins. Okay, right. So basically, the picture that I am about to draw you is exactly the same as for the classical cadherins, but I'm going to redraw it anyway. Okay, so here is the phospholipid bilayer. Okay, here is the membrane-spanning alpha helix. Then we have our five extracellular cadherin domains. Okay, so desmosomal cadherins have a very similar structure to our classical cadherins, basically. Okay, and here is the amino group right over here. Then we've got, oh, and this is the other thing that they share in common. Again, it's going to be the N-terminal extracellular cadherin domain, which is going to interact with the extracellular cadherin domain on the other desmosomal cadherin. Okay, so once again, here's the amino group, and then we've got 5 extracellular cadherin domains here. Now, I won't draw them all. Okay, you get the message that there'll be 2 more there, and then it will be have its membrane-spanning alpha helix on the next cell. Okay, right. Then we've got our C-terminus here. Okay, and here's the carboxylic acid terminus itself. Right, so let's add some colour on here. So let's colour in the extracellular cadherin domains in orange here. Okay, so all 5 of them here. Okay, and once again, these extracellular cadherin domains will have these hinge regions in between them. Okay, one here, one here, one here, and one here. And those will have calcium binding sites within them. And basically, if calcium binds there, the thing becomes rigid, whereas if calcium is not bound there, it will be very flexible and it will be flapping around. So only in the presence of extracellular calcium can these things actually form this link with another desmosomal cadherin on another cell. Okay, we'll then colour in the membrane-spanning alpha helix here in red. Okay, right. Now, what are the key examples then of desmosomal cadherins? Well, the key examples are the family of desmoglines. Okay, and there are 3 desmoglines. Oh, sorry, not 3 desmoglines. There are 4 desmoglines. There is desmogline 1. There is desmogline 2. There is desmogline 3, and there is desmogline 4. Okay. And then there are 3 desmocolins. Okay, this is the other key family of desmosomal cadherins. Okay, there is desmocolin 1, desmocolin 2, and desmocolin 3. So all of these 7 desmosomal cadherins that we have here, grouped into these 2 families, they all look like this, and they can form interactions which will either be homophilic interactions, okay, in which basically you join let me just discuss this in a little bit more detail because it's a little bit more subtle in the case of these desmosomal cadherins than it was for the classical cadherins. Okay, so basically in this case, homophilic binding means that the desmosomal cadherin is going to bind to another desmosomal cadherin that's in the same family as it. Okay, so basically you can take 2 different members that are both within this desmogline family of desmosomal cadherins, and if you bind those 2 together, that would still be considered homophilic binding because they are so similar. Okay, they're in this same family of desmosomal cadherins. Okay, and it's the same for desmocolin. Okay, if you take 2 different desmocolins, okay, because they're in the same family, if you bind those together, it's still considered homophilic binding. Okay, if on the other hand you were to take 1 desmogline and bind it to a desmocolin, that then would be considered heterophilic binding. Okay, because those 2 are in different families. Okay, so there wasn't that complexity for E cadherin or N cadherin or P cadherin because they weren't families, they were just individual proteins, whereas these are whole families. Desmogline is not just 1 protein, it's got 4 members, okay, and desmocolin has 3 members, okay. So if you've got 2 that are in the same family of desmosomal cadherins. Okay, so they're both desmoglines. That would be a homophilic bond if you had a bond between the two. Okay, right. So that's the formation of these interactions between the two cadherins then. Okay, we now want to look at the machinery that's attached to the cytoplasmic tail, which is how the desmosomal cadherin is going to attach to the keratin filaments, or the other sorts of intermediate filaments within the cytoplasm of the cell. Okay, right, so let's look at the proteins which are going to bind. So once again, two proteins are going to bind to the cytoplasmic tail. Okay, like so. Now, these two proteins are called Placa Globin. Okay, so we'll have this one here as Placa Globin, and the other one is known as Placafilin. Okay, so you have Placa Globin and Placafilin. Okay, and these are both attaching to the intracellular cytoplasmic tail of these desmosomal cadherins, so either a desmogline or a desmocolin. Okay, so we'll have Placa Globin here in vivid purple. Okay, then we'll have Placafilin here in turquoise. Okay, then another protein is going to come and bind on top of the Placa Globin and the Placafilin, like so. Okay, and we'll have this protein in red. Okay, so this protein that's binding on top of both of them, okay, this is called desmoplakin. Okay, and now the desmoplakin then is going to attach to the intermediate filaments. Okay, so this then is going to attach to one of these intermediate filaments. And the good example of an intermediate filament then is keratin. Okay, so that's how these desmosomal cadherins that are interacting with other desmosomal cadherins on other cells are going to actually anchor to the cytoskeleton of their own cell. Okay, so this is an intermediate filament. And this is what gives these cell-cell interactions some actual credibility, basically, because if they were just sitting in the cell membrane, you're not actually forming any sort of an interaction between the two cells at all. It has to be between this rigid cytoskeleton of the cell and the rigid cytoskeleton of the other cell. Okay, right, so this is an intermediate filament we've got here. Okay, right. And once again, what will happen is these desmosomal cadherins, these interactions between two desmosomal cadherins, you're not just going to have one of them. Okay, so if we've got our two cells here, okay, let's have one desmosomal cadherin here and a second desmosomal cadherin here. You do not just form an interaction like this where you have one desmosomal cadherin on one cell interacting with another on the other cell. Instead, you have loads of them in the vicinity. Okay, and I will once again stress that heterophilic binding is very common. You'll often get desmoglines binding with desmocolins. Okay, so you don't just have homophilic binding in the case of the desmosomal cadherins. And one of these clusters of a huge number of individual interactions between two desmosomal cadherins, okay, this cluster is known as a desmosome. Okay, so basically the classical cadherins, they cluster together to form these adherins junctions, which are attached to the actin cytoskeleton. The desmosomal cadherins, they aggregate together to form these desmosomes, which are attached to the intermediate filaments within the cytoplasm of the cell. And a good example of an intermediate filament is keratin. Okay, so that now concludes our discussion of the Cadherin superfamily.